Hello, welcome everybody. I'm Sandy Williams. I'm the Marketing and Product Manager at Scientific Bioprocessing. And today I'm going to be moderating this joint webinar between SBI, Phase Holographic Imaging, and BioSpherix. Before I introduce our three speakers, I'd like to go over a couple of housekeeping items. The webinar is being recorded, and you will be receiving a recording after the live event. And also you see in your control panel, there is a chat question function. So please feel free to submit your questions at any time during the presentations. We're going to have our speakers address all questions at the end of the three presentations. So with that, I'm very excited to introduce our first speaker, Dr. Kirsty Alm from Phase Holographic Imaging. And she's going to be talking to us today about a very cool way how you can get non-destructive imaging of your cells real time. Kirsty?
Thank you, Sandy, for introducing me. The next 15 minutes, I will talk shortly to you about why the choice of experiment conditions is important for in vitro drug development. Also, how our amazing HoloMonitor works and how it fits into that workflow with cell-based experiments in research or biotech or pharma. And I will give you a taste of the results that the HoloMonitor can produce, especially for in vitro drug development. So, can we trust our cell-based assay results? To create a cell-friendly environment, standard cell cultures are kept in incubators with 5% carbon dioxide, 37 degrees Celsius, but uncontrolled oxygen. And neither of these parameters are treated with care. Cells are taken out of the cell culture incubator for sampling and viewing, and the incubator door is often open, causing fluctuating temperature, fluctuating oxygen levels, and shifts in pH. Not only you, but also your colleagues will contribute to this. For experimental results to be relevant, they need to be created in a stable environment. Otherwise, all results will depend more on the environmental changes than on any actual substance, cytotoxicity or effects. So in this webinar, we will show you alternative experimental workflows that will address ways of circumventing these environmental issues. For example, SBI sensors are a great tool to analyze the pH and the oxygen levels in cell samples in the incubator. And you will soon hear more about these sensors from Dr. Boy at SBI. So, standard cytotoxicity assays demand that the cells are taken out of their preferred environment quite a few times. Every sample will be affected by this. So, a really common workflow for experiments could look like this. So you prepare for the experiment, you look at your cells, you take them out, you seed them. Then you check if the cells are ready. You introduce different treatments, and then you start your incubation. And during the incubation period, you will remove samples intermittently for analysis. Every time you do that, you will disturb your cell cultures, the remaining ones. And then at the end of the incubation, you still have data analysis to go through. And in addition, if at the end of the experiment results were not sufficient, you need to create new experiments. So samples spend quite a lot of time outside the incubator during this, or disturbed in the incubator. But what if it would be possible to create a workflow that minimizes the time outside the incubator? So a workflow including a HoloMonitor could look like this. So you count your cells using the HoloMonitor, and you can do that inside the incubator. For seeding, you need to take them out in trypsinizing, obviously. Then to check to see if cells are ready, you can use the HoloMonitor in this incubator. Then you need to add your treatments and start the image capture. And then inside the incubator, the entire experiment will run and you can just walk away. And at the end of your experiment, end of the incubation, you get your results as graphs ready to look at. You can already view during the experiment. Because the samples have been treated really well, they have been kept at a stable environmental temperature, and the HoloMonitor does not affect your samples in any way, you can reuse the samples for analysis of, for example, protein content or anything else that's relevant. And in addition, you can take your results from HoloMonitor and reanalyze them. If you did a proliferation assay, you can reanalyze your cells for morphology changes without creating a new experiment, without seeding in more cells. So you will be able to create a non-disruptive, really efficient workflow, making your results more reliable and use up less of your precious substances, and with a lot less effort than you would need to put in with a regular experiment. So what is a HoloMonitor? It is a live cell imaging system. It has software that's a great part of it, the software, that can quantify many aspects of the daily life of a cell. The HoloMonitor is really cell-friendly, and the technology we use explains much about why. So how does it work? We use digital holographic imaging to create 3D images of the cells. Okay, so what does that even mean? The short explanation is that cells are see-through. When light passes through, it will move a bit slower. It will be a delay when the light passes through cells. That delay of light is called a phase shift, and that's what we measure with the HoloMonitor. We capture this delay of light with a sensor. We never capture any cell images. And then we put all that data into a computer, and the computer will reconstruct cells based on the delay of light. So in this picture here, the whiter the cell is, the thicker it is. This is a drawing made by the computer to show the thickness of the cell in each pixel. Beautiful cell image. And just for fun, we can add lots of colors, both to make it more beautiful, but also to enhance details. So all colors and all images you see in this presentation are reconstructions, software-generated images. When using holographic imaging, you don't need any labels or stains. It's all label-free. It's all non-destructive because we have a very, very low intensity light. So no phototoxicity. So you put the HoloMonitor into the incubator, but if you want to do it even better, you can use BioSpherix Xvivo systems, and you will hear more about those in about half an hour from Dr. Han. So based on this technology, digital holographic imaging, what can the HoloMonitor do? Our software is capable of two types of assays. We have the guided assays that will walk you through the setup and the running of the experiment. And the results are displayed while running the experiment. So as soon as the experiment is done, you have your results. You get quantitative results for cell count, for proliferation, for population motility, and for dose response. The in-depth part of the software, those assays are more open-ended. You can extract a lot of different types of results, but more user input is needed. So combining, for example, the tracking with a morphology is quite a powerful tool. So images from both guided and in-depth assays can be reanalyzed for other results. For in vitro drug development, I would like to first focus on proliferation and dose response assays. So usually, cytotoxicity assays, dose response assays are usually their endpoint experiments, and they are often performed with indirect methods, such as MTT assays or XTT assays. So usually data are collected after 24 or 48 hours. Using the HoloMonitor, you can instead collect data at intervals that the user decide, that you decide. every hour, every 10 minutes, every 6 hours, whatever you need. And then data is presented for all time points. And you use the same cell culture for all time points, making the results less variable. So in the image here, you can see that there is a dose response graph, nicely shown with the cell number and the different doses. Then you can also see below that there is a proliferation graph showing the cell number over time. That's the left graph. The center graph shows you a confluence over time. And you can see that those graphs do not mirror each other. There are differences depending on the action of the drug. To the right, the graph shows you the volume of the cells, which is again showing a different pattern than the proliferation data. Now I've added the kinetic dose response graph that the HoloMonitor provides. So at every time point that data has been collected, we have the graph showing when the doses actually take effect. So in short, the HoloMonitor Dose Response Assay is an assay with benefits. You have interactive dose response curves, you have guided assay, you have real-time graphs, and you can reanalyze the data later. Just as an example, I will show you how Dr. Croft and her colleagues investigated the use of the HoloMonitor in cytotoxicity studies. They used 5 different cancer drugs, 2 unknown and 3 well-known, to treat HeLa cells. So the graphs to the right show number and confluence over time, while the graphs to the left show cell thickness versus area. So Croft and colleagues started with cell count and confluence. That's the graphs to the right. And you can see that the graphs for control or untreated, the confluence and the cell number, they follow. But for all the treatments, there is a deviation in the pattern of confluence and cell number. In addition, looking at the morphology, you get further into what happens in your cell cultures. You can see that all the different treatments, in essence, cause cells to become smaller and thicker. So the combination of cell morphology adds value to the cytotoxicity investigation. Cell movement is also important for cytotoxicity studies. Assays for basic cell movement, as well as chemotaxis and wound healing, are important tools. So the movie shows the tracking of individual single cells over time, and the little colored tails show how the cells have moved. So in the image that I show now on top of the movie are the tracks from a chemotaxis assay. So all the cells move downwards, attracted to a chemical, an attractant. If you look at that in a graph, like in the center here, you can see that the cells all move downwards. They have been attracted. They all start at their own 0 point, and they all move towards the same direction. Combined with the cell morphology data for each individual cell, you can, for instance, see if the cells are dividing. In the graphs to the right, you have the volume over time, and you see there are sharp decreases in volume. That's when the cells divide. As an example, I will show you how Dr. Gershkovitz and colleagues correlated tumor cell expression of a calcium channel, TRPM2, I will probably mix those letters up a lot, with reduced neutrophil migration towards tumor cells. So tumor cells usually express neutrophil chemoattractants, quite powerful ones. That expression is regulated by TRPM2. So the researchers have used cell cultures with tumor cells, non-treated or control cells, cells with down-regulated TRPM2, and cells with a restored function. So when they track the movement of neutrophils in these different cultures, they clearly saw that the absence of TRPM2 clearly decreased the attraction of tumor cells. The neutrophils didn't move towards the tumor cells as much at all, but they had a clearly increased motility. So they moved around a lot more, but they didn't have an aimed movement. As you have seen so far, the HoloMonitor fits right into your workflow, as it allows for many basic non-invasive, non-destructive cell investigations, giving biologically relevant data. So use the HoloMonitor as the first point of investigation to study cell behavior or count your cells before you can use that same sample for more invasive or even destructive methods. The HoloMonitor does not only provide lots of data, but it also creates beautiful images and movies. So in these two movies I'm going to show now, the thickest cells are yellow. Keep an eye open for those. So to the left, there is control, untreated L929 cells. And you see when they become yellow, they then pop and divide. To the right, cells are treated with colcemid. You can see they also round up and become quite thick, but they never pop and divide. They just flatten out again. Colcemid will inhibit or will arrest cells in G2, cause a G2 block. So cells will just become larger and larger. The movies show approximately 2 hours for every second that the movie is running. So these cells have been running for 48 hours. And you can see at the end of that, that treated cells are much larger than the control cells. Okay, thank you for listening. If you have questions, please add them to the chat or contact us. And if you want more information on quantifying cell morphology and cell movement, please watch our previous webinars. Thank you for listening. I will now hand you over to Sandy again. Thank you, Sandy.
Thank you so much, Kirsty. That was an excellent presentation. Really enjoyed hearing all the different ways you can use the HoloMonitor, not just for beautiful cell images and videos, but also a lot of assays and data. Excellent. So next, we're going to hear from Jake Boy, who is a senior applications engineer with Scientific Bioprocessing, and he's going to talk to us about optical sensors for cell culture. So let's switch to Jake here real quick. And there he is. Thank you, Jake.
Hello, everyone. Just a second here. Okay, so I'll get started. No, we don't have a whole bunch of time here. All right, so I'll be talking. Oh, thank you, Sandy, for that introduction, first of all, and Shusty, thank you for that presentation. I always love watching those images of the cells. Really cool. So I'm going to be talking to you guys about optical sensors. And first, I'll get started with a little bit about our company and kind of where we come from. So Scientific Bioprocessing, we're a bit of a startup, but we come from a much older, well-established company called Scientific Industries, and they invented the Vortex genie. You can see that little graphic of it down there. It's a little blue box that shakes your tubes and mixes things for you. So I'm sure a lot of you are familiar with that. But we have commercialized a sensing technology from Dr. Govind Rao and collaborators at University of Maryland, Baltimore County. And we've taken the sensing technology they developed and really turned it into something very useful for cell culture. So when it comes to sensing, we think there are a few things that you really just can't sacrifice on. The sensors have to be reliable so they can provide real-time monitoring in the smallest to largest vessel. They must provide a way for automatic control, so a way to use the sensing, essentially. And they must be able to maintain relevant physiological parameters at the cellular level, so where the cells are actually growing. And they must be economically remarkable, or in other words, inexpensive. So the FDA, right? This is a really big deal, especially in drug development. Trying to get through FDA regulations and make sure that everything is up to safety standards. And part of this is the PAT initiative, and this is something that we're really aware of, and that I think the industry is becoming more and more aware of. And that's really trying to standardize as many things about your process as you can. And the reason for this is so that you can have consistent, results that you can replicate. And one way the optical sensors really fit into this is that these sensors can be used in really any type of culture vessel, any type of experiment. They're very versatile and can be well adapted to lots of different scenarios. So where does this fit in with the FDA and the PAT initiative? Well, you can use the same sensors and the same sensing system all throughout your experiment for everything that you do. So there's no switching in between different types of sensing modalities, and there's no training that needs to happen as you switch between different types of sensors, because you can use optical sensors, and that part of your experiment is going to be standardized and it's going to be the same every single time. So where do optical sensors fit in? So typical electrochemical probes just really don't work when we're talking about small culture devices like tea flasks, petri dishes, shake flasks, and also microfluidics devices. So optical sensors can be used in these small culture vessels where previously we could not do real-time inline sensing. And what we see here is just a little bit of data collected using optical sensors in petri dish. These are petri dishes growing cells in CO2 incubators. And here we can see real-time pH monitoring for the duration of this run, something that you could not do before. Introducing the ID Developers Kit. So this is our most basic product offering. And really I'm showing this here because I want everyone to have some context about what I'm talking about, what everything looks like. And you can see here we have the reading devices. They're the black coaster-shaped things that are sitting under the culture vessels. We have a tea flask, multi-well plate, shake flask, and then an evaporating dish. So you can see there's a variety of ways that we can use the sensing type, and there's even more that I'll get into a little bit later with fiber optics. So a little bit about the sensors and what they're capable of. So optical sensors, they have great response time. They come pre-calibrated. This is a really great thing about them. So we calibrate the sensors in our lab. Recalibration is never necessary. So once they're calibrated, you use them. You don't have to worry about doing that yourself. They're meant to be single use. So after a run, you don't have to clean it. You don't have to recalibrate it. You just throw it away and then use a new one for the next run. The sensors are really, really tiny. So they can, so here on the spec sheet, we have the standard dimensions as 1 centimeter squares, and they're 0.3 millimeters tall. So a few pieces of paper stacked on top of each other about that height. And then imagine basically a sticker, and that's what the sensor looks like. But they can be as small as 3 millimeters in diameter. So you can put these sensors really anywhere you want to. And I'll show a little bit of that coming up. So microfluidics. So organ on a chip and in vitro drug development. This is a great area. So organ on a chip experiments are reproducible. They're really low cost and they're high throughput. So a lot of drug screening can happen. And the best part about it is that these can be human tissues that you're using in these organ on a chip devices. So as opposed to animal models, we have a high throughput way to test drugs and screen drugs, things like toxicity, absorption, all of those types of things, using the human cells that the drug will actually be used on eventually. So it's really provides reproducible, more accurate data. And our sensors can fit into these devices in a few different ways. So we've identified 3 main ways, and so I'll just run through those really quickly. So in the channels of the actual organ on a chip device itself, the sensors can go in those channels. They're down to 3 millimeters. Oftentimes, the channels or the reservoirs in the organ on a chip devices are between 3 and 5 millimeters in diameter. So the sensors fit really well in there. Also in the perfusion lines. So we know that in microfluidics, it's really common that you'll have a constant flow of media going through. So you have a perfusion loop or a feeding loop or just media that's circulating. And we have a flow-through cell that houses a sensor that can fit in these media lines, in these tubes. And then also in the media reservoirs themselves. So if you have the media that's being fed to the organ on a chip device, we can have sensors in that media. We can also have sensors in the waste media. So here's what an organ on a chip setup looks like. And then you can see there's a few places that I just mentioned. There's the media reservoir there, and that little vial with the media that you see. There is the perfusion loop and the tubes, and then there's also the reservoirs and the channels in the chip itself. And here's what that looks like. So this is an organ on a chip customer that we're working with, and you can see these are the 3 millimeter pH sensors. They're the little white spots that you see in the reservoirs in this organ-on-a-chip device. So a couple characteristics that make these sensors really, really good for these applications. First of all, they can be really small, like you can see here, and they only require a 10 microliter fill volume. So we're still maintaining that micro level that coins the term microfluidics, and you don't have to adjust what you're doing. You can just put the sensors in the chip and operate as you normally would. And then this is read using fiber optics, which I'll show a little later in the presentation. So now I'll talk a little bit about the T-flask, something that everyone is familiar with. And I just want to talk about some common, misconceptions or some cell culture myths, if you will, about the T-flask. So here's a study using two T225 flasks in parallel in a CO2 incubator. And the researchers wanted to test, one of these common practices that people who use T-flasks, employ. And I used to do this as well when I used to do cell culture. We had T-flasks stacked up in the incubator, and we thought, hey, if we crack the lid, it'll help the cells become more oxygenated because it'll let in the environment in the incubator. But, this is just something you couldn't really test. You just thought logically this would work. The researchers here, using optical sensors in these T-flasks, were able to monitor the oxygen levels and really put this theory to the test. And what they found is that in the partially open T-flask and in the closed T-flask, it really didn't matter. After about 60 hours, between 2 and 3 days, the T-flasks became completely anoxic. And the partially open flask, after another 3 days, did build up a little oxygen eventually, but still very hypoxic compared to where it started. So the point here is that optical sensors allowed these researchers to put something to the test in cell culture, and they were able to find that actually our T-flasks are becoming hypoxic. So what the researchers determined is that T-flasks are mass transfer limited. So the KLA, the transfer of that gaseous environment in the incubator does not transfer through the liquid media down to where the cells are actually growing and metabolizing. So the T-flask is mass transfer limited, and the authors thought, okay, if we gently agitate the media, break up that insulating layer a little bit, maybe we can get the oxygen down to the cells. So here's the experimental setup. Figure A, you can see the static T-flasks. These are T75s. And then also you see below that in the same incubator, another T75 on a rocking platform. In figure B, you can see the optical sensors sitting stuck to the bottom of that T75 flask. So they measured pH and dissolved oxygen as this rocking platform agitated that media in the hopes that it would allow the gas to reach the cells as they're growing. What the researchers found comparing the static flask to the rocking flask was really astounding. And I'll dive into these figures here. So figure D, this is showing the pH. In red, we have the rocking flask. In black, we have the static flask. And what you see is that agitating the media allowed the cells to maintain closer to physiologically relevant levels of pH throughout the duration of the experiment. And part of the reason why this is happening, when cells metabolize, when they respire, they're using up oxygen, they're giving off CO2. CO2 is heavier than oxygen, it has that extra carbon, so it sits on top of the cells and it forms this little pool of CO2 that starves the cells of oxygen, but it also acidifies the cells because CO2 is acidic. So what you get is this pocket of acidic media right on top of the cells where they're growing. If you agitate that media, you can dilute that CO2, that acidity, and allow the cells to breathe, but also regulate the pH a little better. And what we see here, unsurprisingly, is that the cells that were rocking and were healthier and happier because of this physiological level of pH had a 31% higher antibody titer than the static flasks. Here's figure C. This is looking at oxygen much in the same way. So the rocking flask is in red, the static flask is in black, and what we see here is that the static flask, again, after about 60 hours, became completely anoxic. The flask that was rocking and agitating that media, breaking up that insulating layer, maintained a level of oxygen that never really dipped below 70% for the duration of the experiment. So where does all this fit in with drug development? So there are a few anecdotal examples here that we have, and these are all published in literature that you can go check out in the bibliography later if you want to, but I'm sure many of you are familiar with these things and how hypoxia can affect different things in drug screening. So here there's a study talking about how hypoxia and reoxygenation exacerbates the cytotoxicity because it opens a transition pore in the mitochondria. So here it's just illustrating why regulating oxygen and knowing your oxygen levels during your experiment is so important. And here, another example, hypoxia with an anti-cancer drug, activating that drug in solid tumor treatment. And then here we have, there's another study showing that regulating the pH in conjunction with hypoxia could be a way to fight pancreatic cancer. So it's really, really important to know what's happening with your oxygen and your pH during this drug development stages, so that you don't end up wasting your time, and so that some of the effects that the drug will eventually have, and some of the potential that it could have as well by measuring these parameters. So a little bit about our technology. This is the reading device I showed before. This device is meant to sit under something like a tea flask or a multi-well plate or a shake flask, a petri dish, something like that. This device is meant to go in or out of an incubator. It's sterilizable and watertight. And a little bit about how it works. So here we see a diagram. This is a culture vessel. There are protons, there are oxygen molecules floating around. You see the sensor stickers that are stuck on the bottom of the device there. And then the reading device sends light through the wall of the culture vessel that fluoresces when the sensors fluoresce, and then it's interpreted as dissolved oxygen or a pH reading. And the really, really cool thing about this is that these sensors have nothing that connects them to the outside world. Once they're in the culture vessel, there are no wires or anything like that. You close the culture vessel, you never have to open it again. There's nothing that could introduce contaminants, so it's considered minimally invasive, and it's really easy to use because of this. Now, we know that coaster form factor doesn't work for everything, so here we have fiber optics. And the fiber optics fit into a huge variety of culture vessels, microfluidics being one of them, like I mentioned before. And there are several attachments that we use. So here we have a fiber optic probe. That's basically a plastic sleeve that fits over the fiber optic cables, and you can use this in the port space of something like a bioreactor. It's meant to fit into standard ports, and the amount of real estate, right, is really important when you're talking about port spaces in bioreactors. And this device can do dissolved oxygen and pH in one small footprint. So something really important about that. And then after you're done with it, you throw it away and use another one, so you don't have to sterilize and recalibrate this type of probe. We also have a star adapter, so it's shaped like a little star, so those little arms can grab the curvature of different types of culture vessels. It's meant to stick the end of the fiber optic cable to a culture vessel so that it can locate that sensor sticker wherever it is in that device. And then here we have the flow-through cell, which I mentioned before. This is meant to fit into perfusion and feeding lines, and there's a sensor housed in this flow cell, and the fiber optic cable runs to that sensor. So once you have all the sensing, it's really important to do something with it. And part of that is controlling that environment. And here is what we call the ID rocker. This is inspired by the study I spoke about before. This device is meant to agitate tea flasks as they're growing adherent cells in your incubator. So a way to get around that insulating media layer. So when you grow static flasks, we saw they become anoxic. This is a way to agitate the media so the cells are able to get the oxygen they need to grow healthy. Here's a shaking device that we offer. So this is a pretty standard shaker system, but the magic here is that there's a flask holder, and this allows the shake flask to sit securely on top of the reading device, so that as the shaking is happening, that shake flask is not shifting or moving around on the reading device. It allows for pH and dissolved oxygen measurements in shake flasks. And then we have our suite of software called the ID Data Hub. The software displays the data. It displays it numerically. It also displays it graphically, and it's a dynamic graph, so it updates in real time. And it also allows for some cool features like adjusting the scan interval time, so how quickly you're taking readings. We can go down to 10 seconds, so you can have reading updates every 10 seconds. And this also provides data logging, so you can save your data for as long as your run goes. So we know there's more things people are interested in. It's not just pH, it's not just dissolved oxygen. So we're moving towards getting glucose and lactate added to our optical sensor suite in the near future. So here's the bibliography for anyone that wants to check it out later on. And with that, I'll leave it to your imaginations using some of this new sensing technology and what you can come up with for ways, to find new data and to gain new insights. So with that, I want to thank everyone for listening, and I'll hand it off to Sandy.
Thank you so much, Jake. That was a great presentation. Really loved hearing about how you can place these sensors in the tiniest geometries, like a microfluidics device. That's pretty amazing. So thank you for that presentation. While we're switching to our next presenter, I would like to remind everybody, if you have any questions for any of our speakers today, please just enter them in the question chat box, and we'll be addressing them shortly at the end of the presentations. So next, I would like to introduce our next speaker, Dr. Alicia Henn, who is the Chief Scientific Officer at BioSpherix. And she's going to be talking to us about something pretty exciting. How do you convert your lab over to a cytocentric lab? So Dr. Henn, over to you.
Thank you, Sandy. Now at BioSpherix, we build everything with the cytocentric point of view. To describe this to you, I'm going to start with a short little mental vacation. This is going to be something different. I want you to take a moment and close your eyes, and we're going to build our own private island. So close your eyes, take a deep breath, relax your shoulders down. Now imagine a piece of land. Surround it with ocean to isolate it from everything outside. Give your land some mountains, give it some rivers, some hills, some beaches. Now picture your favorite animal there, whether it's a polar bear or a river dolphin or a flamingo. Now give your island a latitude and longitude for the right climate for that animal. Does your animal need cliffs to soar from? Does it need lush tropical forests? Does it need sand and surf? What kind of food chain do you need on the island to support that animal? What are the right conditions that it needs? All right, now open your eyes, come back to me. Well, what if you could design an island for the cells or tissues that you study in your lab, that you need for your assays? And you need all the tools in that environment to be able to study those cells and get the proper data. So the oxygen, the CO2, the temperature, the humidity would sort of be like the latitude and longitude on your private island for them. What in your lab is designed to actually support the needs of cells? Can it do all that? Now, a lot of people think of this, your traditional room air cell culture incubator. This technology has been around for 70 plus years. I'm going to make the case today. This is actually designed for the needs of people and not for the needs of cells, actually. I mean, it's easy to get your hands in there. It's easy to get your face in there. But if you think about it, it's not the best environment for cells. It's got that water pan in the bottom that's always contaminated, so it's a high risk of contamination. The oxygen levels in there are all wrong for cells that come from inside the body that require lower physiologic oxygen levels. The CO2 and the temperature are fine as long as the door stays shut, but you have to access your cells, and so does everyone else in the lab. So every time that door opens and shuts, all the conditions inside change. The CO2, the temp, the pH, all of this goes off every time the door opens and shuts. So it's a highly variable environment for cells. At BioSpherix, instead of making a people-centric environment, we make a cytocentric environment for incubation, where incubators open only into a closed cell processing area, where you can have full-time relevant conditions for cells. So the gases inside, which are from tanked gases, can be set to the proper oxygen level, the proper CO2 level. They're warm to the proper temperature, body temperature. They're constantly HEPA filtered, so you have aseptic conditions for your cell handling. You don't have open room air or BSC conditions for cell handling. Because in a BSC, it's even worse for cells. Not only is it hypocapnic, it's hyperoxic, and now the cells are cold, and this, of course, is the point at which they're highest risk for contamination. And you're handling them in, of course, completely irrelevant conditions for the cells or tissues. Whereas in a cytocentric cell handling space, you can have full-time relevant conditions. Even the floor is warm to 37 degrees, so that the cells are never even cold. More generally, this module acts as a giant incubator that you can reach your hands into and handle your cells in without them ever having to come out here into people-centric room air conditions. And you can have all the tools you need inside for to study your cells in these cell-relevant conditions. Microscopes, flow sorters. You can have analysis equipment like the IncuSite, the Seahorse, but more broadly, with all this equipment, we can combine into a perfect environment so that when the cells first come into the lab, they go straight into the Xvivo system, and the Xvivo system will protect them across the entire process, start to finish, so that no matter what the cells need, it can be inside the cytocentric environment. Now, this is the Xvivo that I had designed for my work in our lab, where we have 6 different incubators. That are these black doors that you see here. So I can have 6 different conditions at the same time for my assays. And I can use this so that 6 different people have individual environments for their experiments, so that nobody is interrupting someone else's experiment by opening that door to the room. I can also set this for different oxygen levels, so cells never have to be in people-centric room air conditions. So our customers are using Xvivos like this to study cells and tissues in controlled oxygen conditions to better simulate different environments, like this study where they've established a placental barrier model under physiologically relevant oxygen conditions, or this one where they're looking at blood-brain barrier model in brain spheroids, or CNS injury with 2- and 3-dimensional cultures, or the human colorectal cancer microenvironment with 3D tumor stroma co-cultures. All of these are physiologically relevant models for drug development. We also have customers that are actually using the physiologically relevant environment to develop hypoxia responsive drugs for cancer treatment. Now, the reduced risk of contamination in the Xvivo system also allows for reduced or eliminated antibiotic usage, because as we know, mammalian mitochondria are evolutionarily related to bacteria. So when you have to constantly flood your cell cultures with antibiotics because of the contamination risk that comes with handling them in room air, you also you can get rid of that risk by enclosing the system and keeping that contamination risk at bay. So you can get rid of the antibiotics. We've been culturing for almost 10 years without antibiotics, and we have not had a major contamination issue yet because of the closed system for cell handling. So let's look at traditional cell culture best practices. You know, you routinely subject cells to swings in oxygen, CO2, and temperature. I'm going to refer you to the other 2 talks in this series for more information about the benefits of unbroken physiologic oxygen. Today we're going to talk more about unbroken temperature and edge effect specifically. So do unbroken temperature conditions make a difference in cell-based assays for drug development? We did a study using PHI's HoloMonitor M4 inside the Xvivo system, looking at edge effect and cell plating temperature specifically, because there was a paper that came out in 2005 that said if you just leave your cells in room temperature for 1 hour after plating them, that you could help get rid of edge effect. But of course, we know this is terrible conditions to be leaving your cells out in. So we wanted to see if we could get rid of that edge effect in a better way. So now we've known about edge effects since the 96-well plate was popularized back in the 1970s. Cells cell non-randomly with cells depositing near the edges of the plate in the wells on the outside edges. It even affects some wells on the next row in. So researchers routinely fill the outside wells with buffer and just exclude those wells from the experiment. And this loses over 1/3 of the turf that you have in your assay. And this is going on in thousands of plates every day across the world. That adds up to a huge waste of plastic and fluids and time. So we decided to set up some experiments where we set up cell plating using A549 cells in either room temperature BSC conditions or in constant conditions in one of our chambers. And here I'm going to switch and show you some videos of what the thermal imaging camera actually reveals when you plate cells this way. So what we see here is on the left, these are room temperature conditions where you see all the bright colors. And on the right, we have constant 37 degrees. We're here where the plate and the tips and the reservoir and the medium and the pipetter and everything is at the same temperature. It all looks the same color. And this is 37 degrees. That's the top of our temperature range. So over here, on the left, the medium was actually warm to 37 degrees before we started, but you can see that it's already cooling even here in the reservoir. And as the cells get plated, they cool rapidly in the wells, and the plate starts to warm up a little bit. I'm going to speed this up a little bit so you can see. Here we're at about 1 minute. The plate is full, and already these cells are being subjected to temperature changes. They're getting cooler and cooler. I'm going to speed this up a little and show you that by 4 minutes, this plate is already cooled to room temperature. So what happens when you put that in the incubator? Here's our 37-degree plate that's been plated at 37 degrees. It's at the same temperature. Here comes our room temperature plate, which is cooler. Now, as that plate warms up in the incubator, you're going to see it warm up from the edges of the plate inward. And all of these temperature changes are happening during the critical time when cells are settling down in the medium onto the bottom of the well and get distributed on the bottom of the plate. So next I'm going to show you what's happening to the cells. This is with the HoloMonitor. Before I start this, I just want to say these are two wells on the right-hand edge of the plate over here. On the, and these are wells F12 and E12. And on the left-hand side, we have a plate that was plated at room temperature and placed at 37 degrees. So it's undergoing these temperature shifts that we just saw. And on the right-hand side, we have ones that were plated at constant 37 degrees. So I'm going to play this, and you're going to see what happens is that with the HoloMonitor, you can see these cells are being rolled across the well. I'm going to back that up and start it again. Rolled across the well during these temperature changes, which might explain why they're distributed non-randomly after they settle down. Whereas when they're plated in constant 37 degrees, they settle straight down like snow falling with no wind, and they flatten out, and then they come into much better focus as they flatten out. And we were even able to show that if you inverted these temperature effects, where you plated cells at 37 degrees and then you put them in room temperature, you could actually make them go back the other way. So that's what we see here on the right. Our cells being exposed to the opposite temperature changes, you can actually make them roll in the opposite direction. So I'm gonna quit this video and come back to my slides. With their cell tracking software, we could show that these cells have a combined vector that's very strongly directional in these edge wells. And with a constant temperature for both plating and incubation, you get rid of this. You get rid of this edge effect. And most of this variability is introduced in the first 10 or 15 minutes of plating. And you can see that we get rid of this again with constant conditions. So that variability starts in the first 10 minutes in the incubator when plating conditions are not controlled. And so which helps confirm this thermal model of edge effect. And here's just to show you what that plate looks like. After you stain the cells, you're able to get rid of the edge effect when you plate them in the proper conditions. So even if you think about the pinnacle of cell culture technology, the cleanest clean room in the world where you could do your drug development, cell-based assays, you still have the same source of variability as the most basic labs anywhere in the world. You still have temperature swings oxygen, CO2, pH level swings whenever cells are taken out of a room air incubator. And actually, it's even worse in a clean room, because if you think about it, these people have to dress up in all these layers of clothes, so they keep those rooms extra cold, which is even worse for the cells. It's essentially a people-centric environment, not a cytocentric environment. In a cytocentric environment, you can have no suboptimal transients, full-time control of cell conditions wherever the cells and tissues need to be. So you can take that environment even, and you can clone it, and you can put it anywhere in the world. So now you can have verifiably the same conditions anywhere you need to do your cell-based assays, whether it's San Diego or Stockholm or a ship at sea, it doesn't matter. It can be anywhere, and you have definitely, verifiably, and recordably the same conditions. So when we talk about scientific reproducibility and about the billions with a B dollars that are wasted every year on irreproducible data trying to develop drugs that can help people, we talk about factors that introduce variability into cell-based assays. We talk about problems with reagents. We talk about reporting issues. We talk about experimental design. We talk about cell identity, and all these factors come into play to different extents depending upon whether it's someone in the same lab trying to reproduce their own work, whether it's a collaborator down the hall trying to reproduce your work, or whether it's someone halfway around the world picking up a paper and trying to reproduce that work. But the one factor that adds variability into every single cell-based assay done on this planet is cell conditions. And we have to get this into front and center in all the discussions that we have about scientific reproducibility. I invite you to my LinkedIn group for in vitro reproducibility, where we talk about all of these things and more. So you can't figure anything out by plunking a flamingo down in the middle of Philadelphia. It's the wrong environment. We have to have these assays in a relevant environment full time in order to be able to get the right answers. So with your own private island, you can put your tools inside, like the holographic monitor and also SBI's ID Rocker. We're starting some collaborative experiments with them as well to see what we can do combining these technologies together for the right conditions for the cells that are at the heart of what we are all doing. So I will stop here and send it back to Sandy. Thank you very much.
Thank you, Alicia. That was a lot of fun. And I love that mini mental vacation you took us there at the beginning. And now I cannot stop picturing that flamingo in Philadelphia, because I'm looking outside my window and we have snow in Minneapolis. So no, it would not do very well. Thank you for really highlighting how relevant conditions can really help with relevant answers in cell culture. I would like to ask all our presenters if you could please turn your cameras on for our question and answer part of the session here. Thank you. So we have a few questions that have come in. So I'm going to start with Kirsty. The first question here is, what is the working volume of the HoloMonitor?
The working volume, I think you need to clarify that a bit.
Yeah,
Does the question say anything else?
No, I would assume maybe the working space, how much space there is to insert a culture plate. But the requester could, if you want to clarify further, please resubmit.
Yes, we can use any kind of standard cell culture vessels like 6-well plates or 96-well plates or Petri dishes or EBD channel slides. So the span where you can put stuff in is about this much.
Okay, so hopefully you all can calibrate how much that was. I'm gonna guess maybe 3 to 5 inches. I don't know if that sounds, or in centimeters. So I'll move on to our next question for Alicia. You have 80% as traditional incubator oxygen. Isn't room air 20% oxygen?
Excellent, excellent question. There's a broad misconception out there that room air incubators are at room air oxygen. You know, this is 20.8% at sea level outdoors, but inside buildings, it's a little lower because of our sealed up buildings. But in an incubator, you have all that CO2 that's being added in, and you have all that humidity, which takes up space in the gas. If you remember chemistry, you know, that means oxygen necessarily has to be reduced. And so we did some experiments where we went around and we actually measured incubator oxygen in a number of incubators across upstate New York. And we got answers that were anywhere between 15 and 18% oxygen, depending upon how recently the incubator door had been opened. Because that opening of the incubator door, because it prompts the incubator to charge back up the CO2, and then the oxygen, of course, is going down if the CO2 is going up. Depending upon how recently that door was opened or shut, you could get anything between about 15 and 18%. If you leave the incubator door shut for 3 days, you can get what will be calculated at 100% humidity, would be about 18.6 oxygen, and that's the maximum you will get in a room air incubator. It's still super physioxic. It's way higher than what you would find anywhere inside the body, but it's not room air oxygen either.
Great, thank you. Next question is for Jake. How do you take readings from a microfluidics device when the sensors are attached to the chip?
Right. Yeah. So there's a couple different ways that we can do this. I mentioned in the presentation the fiber optic method, but we have people who do it 2 different ways. We have someone who we work with using organ on a chip device who sets the organ on a chip device directly on top of the what's essentially the coaster design, and it just so happens that his reservoirs are exactly the distance apart that the two reading channels are on the reader, so he can do 2 sensors at a time doing it that way. We also and more commonly use fiber optic cables, and so the cables come from the reading device, they carry the light, and then you can put those cables right on top of where the sensors are situated within that organ on a chip device. And we have specialized devices we use to sort of hold the organ on a chip device and then fix the fiber optic cables to it so nothing's moving around as sensing is taking place.
Great, thank you. Next question is for Kirsty. They're referring to the slide titled Chemotaxis and Cell Tracking. And the question is, how was this chemotaxis experiment set up? Do you have examples of chemoattractants you have used? And how do you add these attractants to the cells or vessels? Before you start the imaging or during the experiment?
That was a lot of questions bounced up in one. For our when we just try things out to see if they work, we actually use serum starved cells, and then we add fetal calf serum as the attractant, because that's the most, would call it, cell-friendly way of doing it, and it's quite easy to handle. So we usually starve the cells for up to 4 hours, and then we add the fetal calf serum. We use the EBD chemotaxis slides. There you can add your attractant in one well, and you seed your cells first, and you add the attractant, and you have the possibility to have 3 parallel samples where you have a positive and a negative control and things like that. I really recommend the EBD slides for this, or the EBD chemotaxis chambers.
Wonderful,
Did
thank you.
Answer all the questions?
I think you did, but the requester here, you can resubmit if we've missed anything, but I believe you have. So next question is for Alicia. Can I just let cells sit in the biological safety cabinet for an hour before I put them in the incubator to avoid edge effect?
Well, you could, but that's a very stressful environment for the cells in that, it's too cold for them. The oxygen's too high. There's no CO2, so the pH is going off. So I wouldn't recommend it. Your cells might survive it, but they're very stressed. And if you're trying to measure something that's physiologically relevant, you're not going to have the right conditions.
Great, thank you. Next question is for Jake. Do the sensors need to be recalibrated during a 4-week culture experiment?
Yeah, that's a great question. So the answer is no. our sensors come pre-calibrated and they never require recalibration. So they remain very stable, and they don't get fouled up by things like proteins or any metabolites that are floating around in the media after the cells have been growing there for a while. So that's a really nice feature about them. So yeah, no recalibration is necessary, and a 4-week run is really no problem with these sensors.
Sounds good, thank you. And our final question is for Kirsty. What are the imaging limitations? How small can the cells be to be recorded properly?
To have any kind of detail into your cells, I would not recommend anything less than 5 micrometers in diameter. So we can see smaller things, but we can't really measure or analyze for details. So smaller cells can be tracked and counted, but not analyzed for details. So 5 micrometer and up, and you can get some morphological data out of it, as well as the tracking and the cell counting.
Excellent, thank you. So that was the last question. I would like to thank our speakers for 3 excellent presentations, and remind our audience that this is the 3rd presentation in a 3-part webinar series. So you will be getting the recording for this webinar, but then also access to the previous 2 presentations if you would like to learn more about the 2 other topics, which were cancer research and stem cell research. So with that, thank you so much for attending this webinar, and I hope everybody has a great day. Take care.
Bye.
Bye.